Okay, so today we will be beginning with chapter 3b, and obviously starting with section 1 of chapter 3b, which is cell cycles. Where is the whiteboard? So let's put it down over here. Have I adjusted this? I haven't adjusted this. Let's adjust this before proceeding. Right, okay. All right, chapter 3b, the cell cycle. And so under this topic, we will first talk about the process of cell division or cell replication, which is formally known as mitosis. Now, why do you want cell division or cell replication? It's the same thing, right? Biology is a subject where division and multiplication mean the same thing. So you would want this to happen for growth of the organism and for repair of damaged cells and tissues. And right, so we know that organisms as a whole right replicate. It's not exactly replication, right? They reproduce, but that's also happening on a cellular level. Now we're not talking about meiosis and sexual reproduction at the moment, right? We're just talking about one becoming two. And so what happens in mitosis is you've got one cell that goes on to become two daughter cells, two cells, right? And that is how you grow. Obviously, one becomes two, two becomes four, four becomes eight, right? And the doubling continues. Obviously, it is not a never-ending process. It is a very controlled process. Right? It happens with all cells during development and early growth, save a few exceptions. So development and early growth, but right when the organism becomes mature, right, many cells stop, right, with the replication, and few continue. And that depends on where those cells are located, what their function is, how much wear and tear they're undergoing, how much growth or repair do they need. An example is right, so during development, your bones, organs, muscles, right, everything's growing. And so all the cells in everywhere are undergoing mitosis. But when you become a mature individual, right, for example, bones stop growing. Right, and they will only perhaps undergo mitosis for repair. So if, for example, you suffer a fracture, right, minor or major, right, and you put the bone back together to join and heal, so perhaps that is when mitosis would be triggered, otherwise not. But then you've got the liver, right, which is also similar, right? It's not growing on its own, but if it is damaged, then it can, as a form of repair, right, undergo mitosis. And then you've got the skin, which is constantly, right, undergoing mitosis. And that is, right, because you shed skin, a lot of it. You shed skin cells every single day. In fact, sometimes you feel when you wake up in the morning or you haven't changed or, you know, sort of made your bed in a day or two, and sometimes you feel that it's a bit dusty. That might, some of that at least might be the skin cells that you shed during your sleep at night, right? So why is the skin constantly undergoing mitosis? Well, that's happening because it's constantly, it is the protective barrier. It is constantly exposed to the external environment. There is wear and tear. It's exposed to water and heat and everything, and so the topmost layer is thick. It is living, right, but then the cells die off due to the wear and tear, and right, new cells underneath take its place. So that's happening all the time. So to the extent to which mitosis continues in different regions of a mature organism's body differs. Okay. Now, right, oh, mitosis is also, right, involved. It is also the process for asexual reproduction. That's one word for you. Right, and that is when one cell or one parent organism, a single parent, right, gives offspring, right, via mitosis. That are genetically identical. Why does that happen? Why are they genetically identical? We'll talk about it. So a bit of, right, detail for this will come later. But yes, asexual reproduction, whether, for example, it's happening in bacteria, single celled, whether it's happening in certain plants. In fact, many plants can undergo asexual reproduction. You use cuttings. So are you familiar with the money plant?
Pardon me? Can you repeat?
Are you familiar with the money plant?
Yeah.
Right, so you know that you can break off a stem from the money plant, put it in a pot of soil or in a like flask with just water in it, and it will start growing from there.
Yeah.
And that is an example of asexual reproduction.
Mm.
Right? Okay. Sexual reproduction involves parents, two parents, and right, the process by which it happens is called meiosis, which will come later. So let's talk about right? Chromosomes. So we know that nearly all cells have genetic material in their nuclei, right? So you've got a cell that has a nucleus that has genetic material, to use a generic term. And in this case, we're talking about the DNA, not RNA. RNA is also genetic material in the process of transcription and translation. So we have DNA in the cell. Now, what are chromosomes, right? The basic genetic material is DNA, right? So you've got DNA, right? And DNA in the cell, in cells that are not dividing, so they're not undergoing mitosis, are loose threads, right? And are not visible via microscope, via microscope. But for a cell that is divided, the DNA starts to coil. And so, right, you will come across terms such as DNA, nucleosome, chromatin, and chromosome. And these are all the same in principle. It's all the same thing. So all of these are actually DNA. What's the difference? They are different levels of coiling and condensation of the DNA molecules. Because DNA is actually very long, right? A single strand of DNA is, right, the length of a football pitch. How long is a football pitch? So if you just look that up, right, it is anywhere between 90 to 120 meters. And so you've got, right, a single strand is anywhere between 90 to 120 meters, because that, the compared to a football pitch, and a football pitch can be anywhere between that much. And then you've got 46 of these guys, right? So you do the math, and that is a lot. And we know that the nucleus is like super small. So that is, right, for a eukaryotic cell, it is 5 to 10 micrometers. Which is like 5 into 10 to power minus 6 meters. So you've got a nucleus that is that small, or you take 10, and you want to fit in, right? If we take the lower end, so 90 times 46 is 4,140 meters. right, of DNA into, again, let's take the higher end of this, 10 into 10 to the power minus 6 meters. And so you can obviously understand it needs to be very, very, very tightly coiled and packed and condensed for all of that to fit into just this. So how does that coiling occur? Right, so we start off with the DNA double helix. Right, so you've got something like that, right? And you've got proteins called histones. That's supposed to be the DNA. And so the DNA molecule wraps around the histone proteins. And what enables this wrapping to occur well? These guys are basic and negatively charged. Right? How does that help? DNA is acid, it's an acid, right? So we've got histone proteins that are basic. The DNA has phosphate groups, right? You've got negatively charged groups over here. And it's also going to interact with the R groups. In fact, no, R groups are amino acids, sorry, mixing that up. Right, with the nucleotide bases. So it's going to interact with the nucleotide bases. And so that's going to help, a solid coiling. So the initial coiling of the DNA around the nucleosome gives, around the histone proteins, right, gives a structure, a dense cluster known as nucleosomes. Now, this is just the very first level of coiling, right? You've got multiples of these nucleosomes, and these nucleosomes undergo further coiling. And then after that, right, they undergo even further coiling, called, where they form a supercoil. And that supercoil is the chromosome that is visible during microscopy, right? And the reason why it's visible during microscopy is obviously it's become dense, and because of that it stains much better. So obviously we've gone over this during microscopy. Stains are used to give color to the structures, and so it is only dividing cells, actively dividing cells, right, in which chromosomes are visible. Because chromosomes only form during cell division, otherwise they're loose threads of DNA moving around. Maybe some level of coiling, but not enough to be visible. So they only reach this level of coiling, only form during cell division. Right. Now, how many chromosomes do we have? So every species has a characteristic number of chromosomes. Characteristic number of chromosomes. Right, some may have 18, some may have 22, some may have, right, just 10, right, some may have 36, so, even numbers, right? Why we have even numbers, we'll talk about that. So humans have 46 chromosomes, right, or DNA molecules. We normally refer to them chromosomes in general, right, even though, as I mentioned, right, it's the same thing. So they're 46, and they're 46 because they're actually 23, right? 23 coming from each parent. And so what happens is that they form pairs, right? So this becomes 23 pairs of chromosomes in humans, obviously. And right, 22 of them are called autosomes. Right, so autosome is the term used for regular chromosomes, let's say. Right, and one pair, which is the last number 23, right, are the sex chromosomes. And as the name goes, they determine the sex of the organism, whether it's going to be male or female. And during the process of mitosis, when when we're looking at cell division, all the chromosomes are visible, and so they mention the karyotype. The karyotype is basically a picture of all the chromosomes of an organism. Karyotype, a picture of all chromosomes of an organism. And if you take a look at the karyotype, the image given in the text, you will note that the sizes of the chromosomes are not the same, right? In fact, you kind of notice a trend that chromosome 1 is the longest, and then they sort of decrease in size, I believe. Yeah. So as you move along, but right, so they're different sizes, they have different information on them, Any questions?
No, sir.
Okay, then let's talk about the cell cycle. All right, let's get into you know what the chapter is about. So the entire process of you know cell division or replication is called the cell cycle. The whole thing, but right, it can be studied as different phases, and that is right. I mean, the cell is not saying I'm going through this phase; it's just going through the process. So, right, we are studying it as we've divided that for the ease of understanding into certain phases. The names you've given, obviously, we are the ones giving those names. And so, right, it all starts with interface. And the interface is also, subdivided into the G1, S, and G2 phases. Right, so it has three different, subphases. For a mature organism, cells that are not dividing, right, are generally in, right. So the book says that all cells, right, non-dividing cells, are in interphase, which is technically true, but to be more specific, they're in the G1 phase of interphase. Right? Why? Because during G1, which is gap 1, right, cells grow and carry out metabolic activity. So basically, they do their job. They function as whatever they're specialized to do. So they're just carrying out their tasks. That's what's happening in G1. So, right, all the cells of your body, all the living cells of your body, obviously, are at least in G1 phase. And because G1 is part of interphase, the book says, right, all the cells are at least non-dividing, actively non-dividing cells are in interphase. Now, right, when the cell, for whatever reason, receives a signal for division or replication or whatever you want to call it, it moves to the S phase. Right? And during this phase, DNA replication occurs. Right? So why do we need DNA replicate? Because you're going from one cell to two cells. Oops. And you want all of these to have the same genetic material. Why do you want them to have the same genetic material? Well, first of all, it is the requirement of a particular cell organism. We just talked about it. For example, any human cell that you pick up, except for the gametes, they will have 46 chromosomes. And so, right, this one cell has 46 chromosomes, and you're going to go to two cells, so you need 46 and 46. You need double, right, that. So that both of these cells will end up having 46. You don't want it doesn't become half that, the two cells have 23 and 23. That's not how it works. For regular cells, again, right, just to, highlight that point. And, right, also because DNA basically controls cellular activity. So you want that instruction manual, that codex in both cells, a complete codex in both cells. And so DNA replication occurs. So you go from 46 to, right, two sets of 46. And how does that happen? So, right, for example, you have this supercoiled chromosome. So this is a single chromosome. Right, and it replicates. And now what happens is this chromosome looks something like this. So, right, it's the same thing. So, but right, so now this is doubled. So this is, how should I put this? One replicate, right, and this is the other replicate, and right, these are now called, this whole thing is the chromosome in the replicated state, and now a single, strand, which is again how much a single chromosome should be, is called a chromatid. So that is a term that you will come across only during cellular division, right? So in its replicated state, one chromosome has two chromatids, right? So 1 and 2. And so this happens for all 46 chromosomes. All right. And then after that, you come to G2, right, where the other, structures and organelles, right, they replicate and the cell volume increases. Why does it increase? Because the amount of cytoplasm is increasing, not by a lot. And so, right, because it's not just the genetic material that you want to be passed on completely properly to the daughter cells, right, but you want the uh, the Golgi apparatus or the endoplasmic reticulum or the other organelles, all of those to be passed on properly as well, right? And so there's a lot of synthesis going on. There's a lot of synthesis going on, and right, ATP is used. Then, right, mitosis occurs. So we'll go into detail of that. And right, finally, the cells split and become two. Right now, there are a lot of checkpoints during the cells, right? So we'll talk about mitosis in more detail because there are sub phases, sub steps for mitosis as well, and then cytokinesis. So, oh, by the way, I just threw those terms. So right? Mitosis occurs after interphase, which is right, the separation of the genetic material, right? So it is the division of the nucleus, right? And so genetic material is separated into two, and then you have phytokinesis, right, in which cells, this actual physical cell division occurs, right, and one separates into two physically. Now this whole thing, it is a well controlled process, cell cycle. So what happens is, right, there are checkpoints and the cell moves from, when it moves, there are checkpoints and the cell moves from one phase to the other, or one step to the other. And there are certain small proteins, right, called cyclins, that are produced, and these bind to enzymes called cyclin dependent kinase, right, or CDKs, the kinases, plural. And so what happens is that the cyclin, which is the protein itself, and this enzyme CDK, it forms a complex, and this phosphorylates other proteins, which cause a change in shape somehow. So changing their shape and bringing the next stage or next step of the cell cycle. And so they've given examples, for example, right, chromatin is phosphorylated, which causes the chromosomes to become denser. So phosphorylation of chromatin, as I mentioned, chromatin is one stage of coiling of the DNA. It is not super dense. It is not at the stage of chromosome, but it is beyond the stage of, DNA strands and nucleosomes, somewhere in between. So phosphorylation of chromosomes causing, further coiling and formation of dense chromosomes. Another example is, phosphorylation of proteins in the nuclear membrane. and this leads to the breakdown of the nuclear membrane. Right, during cell division. Of course, that during cell division meaning, right, the visit about one stage of mitosis, which we will cover, believe, in the next class. Right, so we have completed 3B1, and so tomorrow we will talk about mitosis, and yeah, so we'll definitely complete mitosis tomorrow, and if we have time, we'll see where we go on from there. Any questions?
No, sir.
All clear, all good?
Yes.
All right, so that's all for today's class, and I'll see you next time, inshallah.
Insurance.